Okay, let's get started. So today, it's really a pleasure to introduce Harry McNamara to most of you. Um, so uh, uh, Harry knows the secret handshake. He was a Yale physics undergrad, worked with Rob Sholkoff. Then he went on to Harvard Physics to do a PhD working with Adam Cohen, where he did really nice work building electrodynamic circuits with the components that neurons use, but in cells that don't have electrical dynamics. So he could really understand all the pieces of it. Then he went on to Princeton. He did a fellowship in the Louis Siegler Institute, the same program I was in, as another connection. And he moved here just 1 month ago. So really as brand new as they could possibly be. And today he's going to tell us about decoding and controlling multicellular self-organization in organoids. So this is very exciting work. I'm very excited to hear about it. And let's welcome Harry.
Hopefully, hopefully you can hear me all right. Yeah, it's a real treat to be here. I actually think this room is where I had one of my first classes as a first year in Physics 200 with Paul Tipton back in the day. But so it's great to be back. And yeah, and to have a break from unboxing equipment and figuring out how to work SciQuest and whatnot. But yeah, as Ben mentioned, so I just started in the molecular, cellular, and developmental biology department. My lab is actually downtown in the new Wuxi Institute. And I'm excited to tell you about my program using stem cells and these stem cell organoids as a kind of physics laboratory, in a sense, to understand fundamental aspects of pattern formation and self-organization in living matter. So I realize, okay, hopefully it's a bit of a delay here. I may dispense the pointer and just go to.
Your cursor might be.
It was working before. Let's do a great start. There we go. Okay, let's see if it keeps advancing. Okay, so I realize developmental biology is probably far afield from the usual subject matter of physics club, but I wanted to start by saying that I think embryonic development is just this amazing process. We all began our lives as a single zygotic cell, with just as this zebrafish embryo did, which then has to divide to make 2 cells, which you can see at the top, on top of the yolk. And then to make an animal, these cells have to keep dividing, they have to communicate, they have to process information and make complex decisions about what type of cells to make, what type of patterns to form, and also they have to do some mechanics to actually build the structures that will make something which begins to resemble a fish with, you can see a little eye popping up at the bottom, it's a tail, and these little sort of chunks and the side, which will make the sort of segments of the body that make the muscle, tissue, and vertebra. Now, somehow variations on these basic sort of programs in the sense of self-organization give rise to all of the diverse forms and functions we see in the living world. And in the biology community, we're getting increasingly good at describing the complexity of this process. We can measure gene expression in tens of thousands of genes and 1 million cells at once. And so we have really good cellular atlases that describe a lot of the details of this process. But in terms of the actual sort of physical principles to which this thing even works, how cells actually build these structures reliably and reproducibly, we still really are learning a lot of the fundamental aspects of pattern formation in biology. To start with some sort of basic introduction about the components of cell signaling. So we know that cells communicate with specialized sort of communication channels we call signaling pathways. So cells will express special proteins called receptors, which stick out of the cell membrane, and they can bind other proteins, sometimes called growth factors or ligands. And when they bind these proteins, these receptors often complex together. And then when they come together, the intracellular part of the receptor does biochemistry. So it can turn on things like gene expression programs to turn that cell into a heart cell or a fat cell or whatever kind of cell it needs to be. Or it can turn on some other kind of cellular response like cell migration or contraction to shape a tissue. So these pathways are super complicated. They come in different flavors. There's a bunch of them with sort of funny names that date back to old mutant phenotypes in fruit flies like hedgehog. And each of these pathways, there's a bunch of different ligands, there's a bunch of different receptors. They can turn on many different effectors. So in general, it's a sort of many-to-many network architecture. And these pathways can also regulate each other. So it has all of the signatures of a complex nonlinear dynamical system. And I don't know if I can move this down here. That's better. And we know that combinations of these pathways, at least in some aspects, encode a kind of information that regulates cell behaviors during development. But the actual language through which these channels communicate information is still largely mysterious. One very popular idea if my slides will cooperate, is that patterns of these signaling molecules provide a kind of blueprint to guide embryonic development. So if you look in the fish embryo around, does this still work? Around the time before the actual morphogenesis starts happening, you find patterns of different signaling molecules. So this one called nodal appears in this graded pattern, which is highest at the margin between the yolk and the cells and then decays towards the top. Another one called BMP has this graded pattern from left to right. And it's thought that these patterns provide a kind of positional information. If cells can measure concentrations, they can figure out where they sit in this concentration gradient. It gives them a kind of coordinate so that they know what to do to build an embryo. And so much of developmental biology has focused on this very sort of top-down, blueprint-oriented view of patterning. But we know in nonlinear dynamics that complex systems can also self-organize patterns from the bottom up in the presence of feedbacks. And it's thought also in other cases that patterns can appear through spontaneous symmetry breaking to generate spatially structured patterns, even in the absence of a blueprint. Now, this sort of qualitative notion of self-organization can be made mathematically precise. Probably the most famous theory of biological self-organization is Alan Turing's reaction diffusion theory of morphogenesis. So Turing modeled these cell signaling interactions as a kind of chemical reaction. And what he showed is that if you write down a dynamical system with a local nonlinear chemical reaction, and well, on the other slides, you can see the diffusive term, and you model a spatial coupling as diffusion, for specific kinds of patterns, you can get, for specific choices of parameters, you can get these special kind of patterns called Turing patterns, which have this sort of finite wavelength and resemble many of the patterns we see in living systems. So this has been a hugely influential idea in developmental biology. But despite decades of effort trying to actually find Turing patterns in real biological systems, it's been extremely challenging to make contact between these theories of pattern formation and real biological examples. Perhaps unsurprisingly, because real embryos are much more complicated than simple mathematical models like this. Developmental processes involve maybe not just one pathway, but several pathways. Cells are not just sitting on a fixed field. They can move around. They change shape. And many of these pathways, which come decorated with the kinds of positive and negative feedbacks you would need for self-organization, also may be pre-patterned by extra embryonic signals. So trying to tease apart what's top-down, what's bottom-up has been extremely challenging. So I've gotten really interested in these organoid systems as new kinds of laboratories to really tease apart at a more rigorous level fundamental aspects of multicellular patterning. So you can take stem cells, these are cells which kind of resemble cells of the early embryo, and grow them in a dish sort of abstracted away from all of the complicated environmental cues they would receive in vivo, but they can still recapitulate aspects of development. This is showing a very incomplete sort of gallery of some examples of these systems. You can get cells to form a little anterior posterior axis, which I'll discuss shortly. You can coax them towards particular organ systems. And you can, for example, send them down a neural path and they make neural tissue, so-called brain organoids. And more recently, groups are trying to make complete embryonic body plans from all grown from stem cells. So it's probably hard to interpret these blobs with all of these sort of letters written on there, but it looks remarkably like a real mouse embryo at that stage. And so I'd say the field has been largely moving towards making these models more and more complex, sequencing them to compare them to what happens in vivo. But I'm really interested in using them, again, as a laboratory to try to understand how cells even do this. How do cells break symmetry to form these types of multicellular patterns? Now, the basic strategy in my group is to use tools from a field called synthetic biology to really interrogate these cell signaling interactions. So if you want to figure out how these things work, I think you really need to get into the logic of cell signaling. And you can use tools from molecular biology to create synthetic genes in cells that can either measure or in some cases record cell signaling interactions, or in other cases control cell signals with light to see if you can decode these sort of programs of self-organization and ultimately test your models with perturbation. And so I've been using as a sort of catchphrase for this approach synthetic developmental biology, both because this field of synthetic biology and programming cells, we use tools from that field, but also in the sort of philosophical sense of trying to understand pattern formation by building things from the bottom up. So I'll tell you, that's it for introduction. I'll tell you 2 examples of what this looks like in practice. First, starting with asking how cells do one of the most important things in embryonic development, which is to form a patterning axis. So early in development, the embryo has to go from being a sort of mass of undifferentiated cells to something which has a head, is going to make a head and a tail. This is an anterior to posterior axis. There's also a dorsal ventral axis. This is the back to belly, and a more subtle left to right axis. And this is, as one famous developmental biologist has quipped, this is a very important thing for the embryo to do. And in vivo, this is thought to be patterned by these extra embryonic cues, which create, again, a kind of blueprint for cells to read out. So this happens in a process called gastrulation. The gastruloid is one of these organoid models which can recapitulate this in vitro. So you can take, it's kind of, in speaking of introductory physics, I mean, we joke in physics, assume a spherical cow. I mean, this is kind of like assume a spherical embryo, but this is a real experimental system which you can make. And you can take a sphere of cells and you transiently activate one of these signaling pathways, a pathway called Wnt, which I'm showing in cyan here. And then this sphere will break symmetry to form a pole of Wnt activity that marks the movie didn't play, which will mark the posterior of the structure as it begins to elongate. So while in vivo, people have worked very hard to describe how all of these extra embryonic cues can pattern an AP axis. I'll use AP as a shorthand for anterior posterior. It was unknown in this system how this axis sort of arises out of cell signaling interactions. So just to give a little more.
Actually, in that experiment, are you
sure
the signal uniformly, or is the signal you're applying also breaking symmetry?
Yeah, so you can imagine, for example, there's radial asymmetry, and then maybe, so the way we activate this pathway is with a small molecule. That was one idea, is that maybe it only activates cells at the outside. I won't get into all of these details in this talk, but when we measured single cell Wnt expression profiles, as far as we can tell, every cell responds to that cue. But there will be other hidden sort of broken symmetries later. Okay, so and please feel free to interrupt with questions at any time. Yes.
So by extrinsic cue, you mean somehow the environment is not spherically symmetric? Or
In the, right, in the embryo, this is, yeah, this little wacky structure is what a mouse embryo looks like. So the salmon colored cells here are the ones which will actually make the animal. And specific regions of these other groups of cells, or sort of extra embryonic cells, they create these localized signals, which are thought to tell, for example, the posterior of the embryo, put the tail here because the extra embryonic cue is doing this.
Okay, but then you have to ask, how did that?
Where did that come from? Yeah. Yeah, so in some cases, ultimately, it usually comes from the mother. For example, where the embryo implants sort of sets the geometry. In fruit flies, they actually sort of deposit the RNA in the anterior and posterior poles. This is just what the geometry happens to look like at this developmental time. Great. So in the gastroloids, what we're here, there are none of these patterning cues. What you do is you do add an extrinsic cue, which is structured in time, but as far as we can tell, it's activating cells everywhere. You add it just for 24 hours, and what you see is this sort of structure shown in magenta here, eventually stops being a sphere and starts to elongate into this little cigar shape. And if you look at signaling activity in this pathway, you see it goes from being absent in the structure to active throughout the structure to now forming in these sort of locally clustered domains before eventually localizing to the posterior, the future posterior, which then elongates. And I should mention that we measure these signals using these sort of synthetic genes, which make fluorescent proteins in specific signaling states, which we can then measure with a fluorescence microscope. So one popular hypothesis has been that this is an example of Turing's reaction diffusion hypothesis, specifically that positive and negative feedbacks in the form of secreted activators and inhibitors of Wnt signaling could take this, initially uniform but noisy sort of initial state and then spontaneously transform it into a polarized structure. Now, this is an attractive hypothesis because it's it ascribes a role to these positive and negative feedbacks in the signaling network. It is consistent with the basic patterns we observe in the gastroloid, and Turing's model is one of the most beautiful and famous in mathematical biology. But these dynamics could also be consistent with other kinds of self-organization. I mean, you can imagine that these cells don't sort of resolve to a single pole by long-range signaling, but it could instead spatially rearrange. And indeed, if you live image this process, so now I'm showing different phases of this polarization process. On the left, first, the transition from sort of, at least at a coarse grain level, uniform Wnt activity to the formation of the first poles. These slides are a little clearer on the sides. And then later, the sort of resolution of those domains into a polarized structure. I mean, you can see the cells are moving all over the place. They're not just sort of sitting in a fixed field communicating with long-range signaling. They're crawling all over the place. They're sampling different interactions with their neighbors. And it's really hard, at least with our sort of available technologies, to actually track cells to really infer what signaling states are over time and to see if we could predict where they end up based on signaling dynamics. So our strategy instead was to ask if we could really identify when we can predict final cell fates based on early signaling states by recording signaling states in cells' genomes. That way we could really identify when symmetry is broken and when there's a kind of fate information, in a sense, in different signaling pathways to predict cells' future fates along the anterior-posterior axis. So doing this required some, yes?
That is, the symmetry is broken, but initially you have a sphere, right?
Yes.
Symmetry is broken always the same way, or
yeah, as far as we can tell, there's no other, like you can imagine maybe the part on the bottom that there's some mechanical interaction that biases the pole there. As far as we can tell, it's there's no like spatial cues that orient where the pole forms. It seems to appear in different spheres in different places. There are other signaling states which we'll later discover, which sort of predicts where the pole forms, but there's nothing in the environment as far as we can tell which is biasing it.
Yeah, there's nothing wrong. Just asking whether this idea about spontaneous symmetry breaking is dense.
Yeah, so I'm being a bit, I'm not being particularly rigorous in saying spontaneous symmetry breaking. So the cells are not all the same. The cells have different signaling states and gene expression profiles. So there are certainly, at the level of physics, broken symmetries already. I guess what I mean is that there is no, the formation of a pole of Wnt signaling isn't following some other sort of long-range patterning cue. It's sort of emerging out of the signaling interactions between the cells. Right, so the strategy to try to identify when we can predict based on cell properties whether they'll end up in the pole or not was to record signaling states using a synthetic gene circuit. So there's some busy molecular biology here, which is not so important to understand the details of, especially I realize some of these genetic diagrams are a little, the shorthand is a little convoluted. What this is showing is, so these little arrows indicate promoters that turn on different genes, and the sequences upstream of them can regulate when those genes are turned on. And what you can do is you can take an enzyme called a recombinase, which can switch in a cell's genome a fluorescent protein from a red fluorescent protein to a green one permanently, which is then sort of inherited by the cell's progeny for as long as they continue dividing. If you place that enzyme under the control of two signals, you require one of these biological signals, in this case Wnt, to drive expression of a protein called a transcription factor. And that transcription factor requires another molecule to gate its activity. In this case, a small molecule, a drug you can add to the media. This whole thing fires only in the presence of both of those inputs. You need both the signal of interest and you need the small molecule. So, you can forget about all of these genetic diagrams and just think about it as a kind of logical AND gate. It records a signal in the cell's genome in the presence of these two signals. Now, the small molecule dependence is essential so that you can query a specific listening window, in a sense, so that when you read this thing out at the end of an experiment, you can actually relate the color of the cell to the signaling state at that early time point, which you defined by adding the drug. So this is a cartoon, but this actually works quite well in practice. These are histograms showing flow cytometry data of GFP expression levels. So at the bottom, all cells are labeled as green in the simultaneous presence of both inputs. Either input alone does nothing. So it really does work like an AND gate. And we can also record with a relatively short drug incubation time, which is helpful to resolve dynamic. I mean, short. It's 1 to 3 hours, which in the context of developmental biology is short. This is the time scale in which these signaling states are thought to change. Okay, so with this sort of technology in hand, what we can do is we can grow sort of cohorts of these gastroloid structures and then systematically vary the time at which we add this drug to label these cyan Wnt active cells green permanently and then see where they end up in the structure. And when you do that experiment, you can generate images like this, where you see it first. If you record before there's any Wnt activity, you don't label much of anything. Okay, that's not so surprising. If you record during the Wnt right after you stimulate this pathway throughout the spheroid, you label cells throughout most of the anterior-posterior axis. Again, this is kind of what we would expect if we think the signal is active throughout the spheroid. But eventually, we start to see some patterns along the AP axis. And in particular, this patchy state, although we don't see an anterior-posterior pattern in the sphere at that time, if we follow those cells with the circuit, we can see them separate along the AP axis. Now, this is just sort of showing that we can do this quantitatively, but the sort of specific structure of the data I'm showing here is not so important. The interesting observation is that this patchy state with these domains of Wnt activity symmetry is already broken. We can already predict where the cells will end up along the AP axis. Now, this was not what we'd expect from at least the most straightforward reaction diffusion model. You'd instead predict that these domains are still sort of fighting it out with long-range signaling. There are other elaborations on a reaction diffusion model, which could be consistent with this. So we wanted to test an alternative self-sorting hypothesis more stringently by, yes?
Actually, I was going to ask, in that picture, the cells that eventually end up, what color are they on the 96 hour one? Are those the blue ones?
Yeah, it's a little confusing because there's a lot of colors. So the color scheme here is cyan is the signaling state is active at that time. Green is the recording. So it actually takes a while for this sort of recording to show up. So the cyan cells are the ones that will be green. The point is, in the final structure, Wnt activity at the end only shows up in the tip. So they don't all maintain Wnt activity the whole time. But if you follow the ones which were specifically active at this earlier time point, you can sort of trace their fates along the AP axis.
What is the unit HAA stand for?
Oh, hours after aggregation. It's just the time base. So t equals 0 is when you form the aggregate of cells. Right. So an alternative hypothesis is that there's a self-sorting mechanism in which these cells are not actually sort of staying in place and changing signaling states at long range, but they actually rearrange themselves to form different domains. But there's a very clear prediction of that model is that in a cell-sorting picture, this sort of ability to predict anterior-posterior fates should be robust to perturbations to cells' relative positions. So you can take these gastroloids and you can randomize cells' positions by dissociating and then reaggregating them into new aggregates. You can sort of scramble cell positions in that way. And in a cell-sorting model, they should still sort out just fine. The cell fates are essentially already baked in. Conversely, in a reaction diffusion picture, to create a new noisy initial condition that would kickstart off a new pattern-forming process that would form a new posterior with a new population of cells. And what we found is it was very clear. So we did the experiment, and we could see these red and green cells still sort themselves out, even following dissociation and reaggregation, which very clearly points to a self-sorting mechanism. We also noticed, sort of incidentally, that due to some idiosyncrasies of the reaggregation process, sometimes you make bigger or smaller aggregates, and their patterns actually scale. So you get sort of a recapitulation of the relative anterior-posterior pattern, even in the smaller aggregates, which is interesting to developmental biologists because you have house cats and you have tigers, and there's sort of interesting questions about how scaling can be regulated during growth. Okay, so this is a qualitative model, self-sorting. This is perhaps a bit hand-wavy. Well, how would cells do this? So cells can also express molecules on their surface which mediate sort of mechanical interactions. So these molecules called cadherins. These cadherins also come in different flavors. Some of them have a homotypic preference. So like cadherins like to stick to like, they don't like to stick to unlike cadherins as much. And when we look at the patterns of expression of these cadherins during this sorting time point, we see that these two cell types express different cadherin molecules. If we perturb expression of these cadherins, we can actually and then create little aggregates, which either do or don't overexpress these cadherins. It actually modulates the interfacial tension between these two aggregates. So we can actually alter the fusing dynamics with this little sort of fusion assay. You can also, if you make mosaic samples where only some cells will overexpress these cadherins, you can cause the cells to sort out. And so we think this is evidence of another very classical theory of developmental self-organization, which is popularized by Malcolm Steinberg, called the differential adhesion hypothesis. And here, the sort of analogy is you can think about these multicellular tissues as like a kind of non-equilibrium fluid. If the cells can move around and exchange positions with their neighbors. And in the presence of differential affinities between cells, cells can sort themselves out to form different structures. And this is how we think at least one component of how we think the gastroloid is generating this AP axis. There's certainly more that could be studied about the actual sort of mechanics and physics of this process. But this was really helpful in helping us disambiguate between these two sort of classical theories about self-organization. Finally, you could ask, and I think several questions have maybe alluded to this. Okay, well, where do these two cell states come from? Are there earlier factors which are sort of contributing to symmetry breaking even before we identify these domains? And if you look at the 48 hours after you aggregate these spheroids, you don't see this signal Wnt at all, but you do see underlying noise in two other developmental pathways called nodal and BMP. So these are not like deliberately activated in the protocol. The cells are just doing this on their own. And there doesn't appear to be any clear spatial organization. I mean, there may be some core shell geometry to the nodal pattern, but we're still not totally sure. But in any case, if you record these early signaling states, you can already predict differential anterior-posterior fates based on even these earlier sort of spontaneous signaling cues. So what this leads us to is a sort of cartoon model of how we think this structure works. When you aggregate a structure of a sphere of cells, it's spherically symmetric, but there is this underlying heterogeneity in cell states. And these differences change how they respond to an initially uniform cue. If you look initially, when you activate this pathway Wnt, it's present throughout the sphere, but some cells maintain Wnt signaling through positive feedback longer than others do. And these differences mediate the cell sorting process through which cells sort out to form a pole. Once you have a posterior pole, I didn't say anything about this, but through relatively well understood mechanisms, this can now drive axial elongation and morphogenesis to create an AP axis from stem cells. Now, if we can ask if we think we understand how these cells make an anterior-posterior axis, can we control it? And so I talked about some perturbations to things like cell mechanics. I mean, these perturbations are kind of coarse. You turn on a molecule everywhere in a structure. If we really want to study the sort of signaling dynamics and language through which these cells are self-organizing, we would ideally like to be able to control cells signaling both in space and in time. So there's a sort of field in synthetic biology called optogenetics, which does exactly this. So you can take these signaling pathways and essentially hotwire them to respond to light rather than to their endogenous morphogens. So this is showing an example of how you can do this. So often this involves taking proteins from things like plants or microalgae that associate in response to light rather than in response to growth factors. And you can use this to cause sort of endogenous signaling responses with light, essentially sort of repurposing photons as morphogens in a sense. So if you sort of design these reagents to control signals in this way and use the techniques, which I don't have to tell this audience, where we're very good at manipulating patterns of light in space and time. Our preferred way of doing this is using digital micromirror devices, so you can play different patterns of blue light. You can take developing systems and actually paint on signaling patterns. This is showing a zebrafish embryo in which we're projecting different patterns of blue light to control this developmental signal called nodal. So a way I'm really excited about extending this work is trying to use this approach to see, can we take these sort of self-organizing systems and put spatial cues back into them to really understand how sort of top-down cues guide and direct bottom-up self-organization. that's all I had to say about that. I think in sort of the last sort of 15 minutes or so, I had some stuff to say about electrophysiology, but I'll pause for questions on that part now, maybe. Yes.
So you mentioned like a perturbation you use is optogenetics, but I know there's also been work where these organoids respond to like physical texture. Have you tried anything with that?
Yeah, I think so. So for example, you can embed, or you think so. In some cases, if you embed them in like matrix, it changes their morphogenesis. Is that the kind of? Yeah. So that so you if you take a gastroloid and you embed it in a substance called matrigel, it actually forms like sort of little body somites. That work is a little hard to interpret because, and it's unclear whether that's a physical, like mechanical cue or something about the signaling factors in the matrix. It's also, you can't really control the spatial and temporal structure in exactly the same way, but there's certainly interesting things to say about mechanical cues and orienting this process as well.
When you were talking about sort of before you got this patchy signaling and Wnt in the cells, the sort of more this heterogeneity and sort of the pre-Wnt parts, do you have like is that just are those differences from like stochastic gene expression or like what?
Yeah. So I'm still working with a student back at Princeton on this now. The short answer is we don't know. So one possible answer is maybe that's a reaction diffusion process, right? Which is on the table. You could also imagine there could be a very short wavelength. Maybe reaction diffusion gives you the salt and pepper, but the sort of longer spatial scale ordering is from cell sorting. That's on the table. One possibility is I'm interested in is metabolism. So I think one way in which the sort of spherical geometry of the structure could matter is that you get metabolic gradients because the cells on the surface have better access to nutrients. And we observe interesting effects where we see more of one of these signals nodal, the bigger you make the aggregate. But that's speculation at this point.
I wonder if the hypothesis that the initial heterogeneity in cell states is caused by reaction diffusion could be tested by, for example, starting with a different geometry instead of a sphere, like having some kind of lobe structure, which would then sort of set the different boundary conditions, right, for this initial state. And could that lead to like different developmental outcomes?
I would need to think about it a little more. I mean, certainly reaction diffusion processes are sensitive to boundary conditions. I mean, you could also imagine that cell sorting mechanisms are sensitive to boundary conditions, right? There can be you can get a length scale associated with them as well. And if you make these aggregates, so one of the popular observations that led people to point to reaction diffusion in these systems is that if you make them too big, you get multiple poles, which falls out of, that's a prediction of reaction diffusion. If there's a finite wavelength, at some scale, you start to get multiple poles. But you can also get that from cell sorting if there's a finite migration velocity and some finite time of the sorting process. So I would need to think about it a little more. Yes.
Maybe a bit related to Derek's question, but like I guess going to the topic of like the heterogeneity in the signaling states before you experience the cell sorting. Like, is it pretty much a guarantee that nothing like there are no cell signaling states that will prevent you from going on to like self-sorting, the self-sorting stage of development? If that makes sense.
Yeah, I don't think there's any guarantee. Right, you can imagine that only some cells. I mean, so one possibility is that like maybe Wnt is just like along for the ride. Like really, there's some other like hidden variable that's like gating cell sorting. We don't think that's the case, in part because you need to activate the signaling pathway to get the sorting to happen in the first place. And they do all still respond to Wnt activity, at least initially. And so that's possible, but I think unlikely.
Do you think that it's side dependent, right? So if you make it too big, multiple.
Yes.
Then what? Then it doesn't cover the top is what?
Well, so it's interesting. The multiple poles can fuse occasionally. There's an interesting preprint from Thomas Greger's group and the Institute Pasteur now, where they make these cache loads of different sizes, and you cut them in different ways. So as you make them bigger, you see multiple poles, which I think is because it can also be explained by a cell sorting mechanism, and you often see those poles sort of coarsen. But if, in some limit, as the size gets too big, sometimes they don't find each other, is a very qualitative answer. I want to say a bit about electrophysiology as well. So maybe I'll jump into a sort of shift gears into talking about signaling with different modalities. And in particular, so usually when we think about developmental biology, we think about signaling with these biochemical signaling pathways, but cells can also communicate with physics, and in particular with electrical signals. So all cells have a plasma membrane, which is electrically insulating and separates the inside of the cell from the outside of the cell, both of which are conducting solutions. And so this plasma membrane has a capacitance associated with it. It can support a potential difference. And that voltage can change as ion currents are passed through specialized proteins in the membrane called ion channels. So you can write, this sort of simple intro E&M circuit diagram for a cell. And an important point is that the sort of conductances associated with these ion channels are nonlinear and voltage dependent. So there is feedback in dynamics because changes in the electric field in the membrane push and pull on charge residues in these proteins, which can open and close the ion channels. And really one of the real triumphs of biological physics and quantitative biology was work from Hodgkin and Huxley, which won the Nobel Prize for showing how nonlinear dynamics in these ion channel feedbacks give rise to the stereotyped shape of the action potential, or the spike. This is the signal that neurons in the brain and cardiomyocytes of the heart use to communicate. And the way this work was studied was using this technique called patch clamp electrophysiology, where you take a sort of fire polished glass and you make a direct electrical contact with the cell, which is an amazing technique, but it's not really, I would say, well suited to studying sort of pattern formation in electrical systems on spatially extended scales. So during my PhD, I was in Adam Cohen's lab, and some of my predecessors in that group developed a way to do electrophysiology with lights. So we called this optopatch it's not exactly a patch clamp, but it's sort of close enough. And what you can do is you can express ion channels which open in response to light rather than in response to voltage to optically stimulate cells electrically. And you can also express fluorescent proteins that sit in the membrane that change their fluorescence efficiency with changes in the transmembrane potential. So we call these voltage indicators because you can use them to read out electrical signals optically. And if your optogenetic actuator and your voltage indicator are far enough separated spectrally, then you can put them into a neuron in a dish and stimulate on the left side and actually watch the electrical signals propagate throughout the cell. So, and actually, this was not my work. This was done before I joined Adam's lab. My thesis research was using this as a tool to study sort of fundamental aspects of electrical pattern formation. So, as I mentioned, all cells have a plasma membrane, not just neurons, not just cardiomyocytes. And you can take a simple cell line, so this is something called a hex cell, which is just something it's very easy to grow in the lab. It's sort of a simple cell culture model. It by itself, it doesn't have any particularly interesting electrophysiology, but if you engineer it to express two ion channels, you can render it electrically excitable. So this cell, if you express two ion channels, a voltage-gated sodium channel and an inward rectifying potassium channel, it will spike. Now, if you further express this optopatch construct, you can both trigger and image these electrical signals with light. So we use these optopatch spiking hex cells to create electrical tissues, which gave rise to collective electrical patterns. So if you grow these cells in culture, they'll form direct electrical contacts with their neighbors. And this is showing a dish with, probably 50 to 100,000 of these cells, and they give rise to these collective waves. You get these sort of spiral waves, which can appear spontaneously in the dish. You can also use microcontact printing to make little electrical or bioelectrical circuits. So the blue is showing where I'm stimulating with a pattern of blue light, so you can launch these traveling waves. These circuits, it's out of the field of view of the microscope, but they sort of snake back on themselves. And you can even make little devices like ring oscillators. So if you print rings of cells and use different sort of pattern sequences to first launch a wave, but if you keep stimulating, you can transiently inactivate the cells there. And then if you poke next to it, you can launch little directional waves because they're blocked in one direction. And if the track length is long enough, you'll see the middle ring kind of eats its tail, but then the outer ones can sort of support these sort of long-term oscillations. So we use this as a kind of sandbox to explore basic aspects of electrophysiology. One thing we used them for was to model dynamic stability. So this is something you care a lot about, especially in the context of the heart, in which transitions between stable pacing and arrhythmia is literally a matter of life and death. Cardiac arrhythmias are a huge problem. People tend to study these and model cardiotoxicity in different drugs by studying specific molecules, ion channels, trying to understand specific aspects of human ion channels and how they differ from mouse ion channels. But what we saw in this sort of model of an arrhythmia in a dish is that the stability transitions between regular pacing and different stability bifurcations, eventually transitioning to pseudo-chaotic dynamics, it depended critically on the geometry of the tissue. So pacing cells sort of synchronously in a sort of 0D mode versus launching traveling waves led to totally different dynamics, which we could really ascribe to tissue geometry because we had this quantitative model, which we could really constrain because we knew every component we had put in the system. But I think the paper from this work I'm most proud of was using this as a way to explore entirely new kinds of, well, I shouldn't say entirely new. I would say unconventional kinds of electrical patterns because electrical tissues are, in fact, a special case of a reaction diffusion system. Presumably, Turing was not thinking about electrical tissues when he wrote down his model. But if you look at the structure of the Hodgkin-Huxley equation, it's exactly the same form. You have a signal whose rate of change with respect to time depends on a local nonlinear term and a diffusive term to capture spatial coupling. And these sort of traveling waves and spiral waves, these are examples of reaction diffusion patterns, which are quite familiar in electrophysiology. But we don't typically think in bioelectrical systems about the spatially structured time stationary patterns of which Turing was interested in regulating embryonic development. So we wanted to ask if we could build a system which would create one of these kinds of patterns. And so the way we did this was by making cells which were electrically bistable. So ion channels can be characterized by their relationship between current and voltage, their IV curve. And you can then sum these up to get if you only have a single cell, the rate of change of the voltage. And we predicted that if you combined certain channels in the right combination, you could put the system into a regime where it actually has two stable fixed points. So usually with electrical cells, you talk about a resting potential, which is the voltage at which the net current is 0 and the cell is at rest. But you can cook up IV curves, which are actually bistable. I should point out there's a historical idiosyncrasy for which there's a negative sign here, which is why the IV curve is flipped. But these are the two fixed points. This is the unstable fixed point. So you can think of this as a kind of cellular electrical switch. And as you change the ratio of these conductances, you can switch between different stability regimes. For example, you can start off in a position where this blue fixed point dominates. As you turn on more and more of this orange channel, you move into a bistable regime until the blue fixed point disappears. And then you're only left with the orange one. And as in many, in all bistable systems, one classic signature of bistability is this sort of history dependence on which fixed point you're at within the bistable regime. If you start on the blue one and come down, you'll stay on this sort of blue manifold. As you go back, you'll be on the orange curve. And so this hysteresis is, of course, a classical signature of bistable systems. So we design cells to do this. And we use as one of the channels, one of these light-gated channels, so that we could experimentally vary the ratio of conductances to switch it between different stability regimes. And this is just showing experimental data of different IV curves recorded in different intensities of blue light. And just to show, they have the same hysteresis effect we predicted from our theoretical model. Now, in tissues, things get more interesting because now you can have domains at different fixed points. And the domain boundary between those regimes has a different stability criterion. It will migrate according to the total area under the curve between the two fixed points. And so we predicted that if we grew extended tissues of this and slowly varied the balance of these conductances, you could nucleate effectively a kind of electrical phase transition in these biological tissues that would sort of nucleate and grow over time. And this is the experiment. What I'm doing here is these are two sort of large patches of cells, about like 50,000 cells each. The sort of pseudo color is indicating the membrane potential, and I'm slowly varying the intensity of blue light. And what you see is you will nucleate these different sort of electrical domains, which then sort of creep along the tissue. And you can sort of pause the blue light intensity at different points, and it'll stay fixed. There's clear analogies to these sort of domain transitions in many other bistable systems. And as we would predict from our model, that the hysteresis is suppressed. So this is a new kind of sort of electrophysiological pattern, but this is kind of a hokey engineered system. So in our paper, we showed signatures of these domains in a stem cell model of muscle development. But the question remains whether these patterns actually play a role in regulating developmental biology. This idea has been around for a while. It's been really hard to test. You can make a mutant of an ion channel in an embryo and you get a weird phenotype. But it's hard to tell, is this a real bona fide effect of electrical signals regulating development? Or is this, you screw up a tissue and you get lots of sort of weird off-target effects? And so this is another direction I'm really interested in continuing here at Yale, is trying to use these stem cell models as a kind of laboratory to test some of these ideas more vigorously. So just in the last sort of minute or two, this is all sort of unpublished data, which I don't know how to interpret yet in fairness. But so if you look at calcium signaling in these gastroloids, there's all sorts of stuff going on. These cells are sort of flickering. You occasionally see domains of multiple cells sort of firing all together. So calcium signals are often downstream of electrical signals and many examples of electrophysiology. Typically, you don't think about calcium regulating gastrulation or anterior-posterior polarization at all, but there's some signal here which could be interesting to study. In another model of gastrulation, this is a 2D model now in which you can get the sort of mesodermal cells to form around the outside. If you electrically hyperpolarize the cells, you can totally scramble the pattern. It's this sort of gene called brachyury ceases from being apparent on a ring around the margin to showing up in all these sort of little spots in the interior. So this is, phenomenological data. It's not totally clear how to interpret yet. I think to really, put this field on more of a solid mechanistic footing, one would want to really establish a mechanism through which electrical signals could interact with some of the developmental pathways that we understand to regulate things like changes in gene expression and cell physiology during development. One potential hypothesis, which I'm interested in testing, is that these signaling receptors in the membrane could also be sensitive to changes in the transmembrane potential in the same way that voltage-gated ion channels have their signals regulated by changes in the membrane potential. Now, this is not something you can measure with a patch clamp, but now I think we have the set of tools to both measure and record developmental signals and to perturb electrical signals to actually scrutinize some of these models. I mean, you can imagine if you measure a dose response curve in one of these pathways, perhaps it shifts and the signal is either inhibited or facilitated with different changes in the transmembrane potential. And I don't know, we'll see, but this is at least a testable and interpretable prediction. So I think I'm just about at time. So yeah, I'm local. I'm here. I'm just sort of setting up my lab, and would love to interact with anyone who is interested in any of these questions. I should mention also, as part of the Wuxi Institute, I'm also interested in starting to explore neural organoids and how electrical signals and developmental signals regulate pattern formation in that context. But I want to also thank you all for your time and for trekking out in the snow. All of the gastroloid work was done together with Jared Tocher during my fellowship at Princeton. The optogenetic control of nodal signaling was primarily with Nate Lord at Pitt, and then oh, I didn't change the text. There's supposed to be a slide of the electrophysiology was done at Adam Cohen's lab at Harvard as a grad student. But yeah, thank you all. I appreciate it.
Do you have any, I know you talked about starting to work on this some, but I'm just interested if you have thoughts now on sort of what you sort of might expect the interplay to be between these self-organized structures and sort of the externally stimulated sort of things that have had a lot more details worked out on previously.
You mean in terms of like in the developmental signaling or the electrophysiology?
I mean, developmental signaling, but I guess it sounds like
Yeah.
Some of that.
Well, so
physiologically.
So I'll give one example. So there's an experiment. There's a science paper from about 10 years ago from the TISAs, which took a zebrafish embryo, and these two signals, nodal and BMP, which I mentioned, sort of predict gastroloid self-organization. If you inject them at the top of a zebrafish embryo, you get an axis duplication. So you get two zebrafish embryos. Their interpretation was one of these classic like morphogen gradient pictures. Is that because it was a very low penetrance phenotype. And their interpretation was 10% of the time you get them just right, and you have to have these precise opposing gradients. But to be honest, I think that interpretation has not been rigorously tested. I think with these optogenetic tools, you can really control the spatial structure and like make a gradient, or maybe you just need some sort of spatial cue to break symmetry and the gradient is dispensable. I think these are the kind of sort of quantitative models of patterning I would like to test, but I don't know, we'll see, we'll have to do the experiments.
Wonderful talk. Welcome back to lecture hall. So for the experiment of sheet of hexol,
yeah,
you observe star waves. Are there some ideas for the pattern selection criteria, or where the singularities are?
Yeah, so it probably comes from conduction defects. So when you grow these in a dish, sometimes there's a little part where they're not as well coupled, and that can create a little sort of a defect that nucleates a spiral wave. It's also thought this is what happens in the heart. If you get like ischemic injury, you get a part of your heart that can't conduct, and then that can nucleate a spiral wave, which causes all kinds of problems.
What is the system? The number of exons?
Yeah, geez. I was like on the order of tens of thousands of cells. So like, yeah. And this is with this, there's some signal processing in there to sort of filter out noise. The signal itself comes from the membranes, but yeah, it's you can think of it sort of like a continuous tissue.
I guess whether voltage could be like a morphogen. I was wondering if that could also have like species dependent, like where the organism lives and the environment it's in. For example, plants are more likely to have like light as a morphogen.
Yeah. So I think Right. I mean, that would be if there was like an environmental electrical signal, I guess, which it's not exactly how I'm thinking about it. I mean, people have done wacky experiments where you put embryos in an external electric field and you can orient things, but that's a little hard to interpret. I think so we know that, I'm thinking about this more about regulating pattern formation within the embryo, not as a response to an environmental cue. But certainly, I guess environmental factors it doesn't have to be the same answer for every organism. One thing that's nice about these stem cell models is you can actually make some comparative studies, is you can grow them from mouse cells, human cells, rhinoceros cells, people are growing organoids from. There's a whole zoo.
If we're in a tax cell experiment, I guess those tax cells are non-accessible, recyclable. So does it matter here whether the cells were accepted or not?
Yeah, I don't know. So in the, for example, in these bistable domains, it's not excitable in the same way. Those cells don't have action potentials, right? I think at least the way I'm thinking about it, for a pattern, when we think about developmental patterning, those patterns form over long periods of time. Embryonic development is much slower than neural information processing. Like a Turing pattern, for example, if one could create, which I didn't talk about, but it's also an interesting question where you could actually make a Turing pattern in an electrical system. You would need a second diffusible component like calcium or cyclic AMP. But a Turing pattern, for example, could regulate development without excitable cells.
Because I have a question, probably you want to solve this, the electric signal propagate between cells.
Yeah, so when you grow epithelial cells, they make these other channels called gap junctions, and they just make little pores between cells, which are not ionic selective in the same way that most ion channels are. So mathematically, it's diffusion.
Actually, I had a question about gap functions.
Yeah.
So you're actually not putting the gap functions in, the gap functions are there?
Yeah, in this context, they're endogenously expressed. You can, overexpress them, and that will change the conduction velocity and the sort of many aspects of the system.
And is there, I guess these links to development seem very compelling that this would be useful, but I guess maybe the two-part question is why, what's the usual explanation for why these need to have gap functions? And is there, what could you do better with electrical signaling in development?
Yeah. So I would say the usual explanation is they don't only, so things like cyclic nucleotides and other signaling molecules, which it's funny. So in the sort of developmental cell signaling world, people talk about calcium signaling all the time. Calcium is known to regulate Wnt signaling and ERK signaling, but with no connection to electrophysiology. So it's possible that these literatures are actually connected in a way which is not understood. And I would say the usual sort of interpretation for why there's gap junctions are to allow for things like nutrient exchange and other small molecules. They're not only exchanging ions. As for what you could do with electrical signaling that you couldn't do with biochemical signaling, I think one thing is actually long range signaling. So a lot of these ideas about long range morphogen signaling haven't really stood up to scrutiny. Well, at least in a lot of contexts, it's actually kind of hard to signal at very long length scales with diffusible proteins. But with electrical signals, you can couple epithelial tissues very far. So there could be a sort of scale dependence. I don't know.
Well, let's thank Harry one more time.
Yeah, thank you all. I appreciate it.